Hello viewers, welcome to my channel. My name is Timothy Kwebelen. I am a clinical research associate. I'm going to continue from where I start in the topic clinical drug development. The phase 1 clinical trial involves 10 to about 90 healthy volunteers are used for this stage of the clinical drug development. Before a volunteer or before volunteer participants in clinical research are being allowed to participate in the clinical trial, they must pass through the healthy check. That means a healthy person is someone who has not got an underlying medical conditions. Somebody who is not hypertensive, somebody who is not diabetic, somebody who is not HIV positive, and all kinds of diseases. For you to participate, you must be healthy. And there are in the protocols inclusive and exclusive criteria. The inclusive and exclusive criteria will tell us the people who are allowed to participate in the clinical trial. And the exclusive criteria also shows us the people who are not allowed to participate in the phase 1 clinical trial. For instance, for you to participate in a phase 1 clinical trial, the protocol of the study must stress and give conditions and requirements for you to be able to participate in trial of this sort. So, what are the conditions? Well, it's phase 1 clinical trial, healthy volunteers, either male or female. So, if it's for male healthy volunteers, that means women will fall under the exclusive criteria. Also, people who are sick and not healthy will fall under exclusive criteria. That means they are not included. Medical students are not allowed to participate in a clinical trial. HIV patients are not allowed to participate in the clinical trials, especially in phase 1 trial that does not involve retroviral drugs or anything that will help the HIV patients. The only way HIV patients can involve in phase 1 clinical trial is where that drug on trial will benefit them. That is either retroviral drugs. Then you cannot test a retroviral drug on a healthy patient. Therefore, you must use a healthy volunteer. Therefore, you must use a HIV patient to do it. If you want to carry a cancer study on phase 1, you cannot use a healthy volunteer to do it. You must use a cancer patient to do it. More or less, they don't have any choice rather than to see if that drug will work for them or not. And before that, or after that, you also have to, in the protocol of the study, design a consent form. Under the medical ethics, you cannot engage any patient or healthy volunteer to participate in your trial without having them to sign the consent forms. And the protocol of the study must dictate what the study is all about. If the study is for antihypertensive drug, you must indicate it that this study is for antihypertensive drug, and you also must show us the protocol indicate the benefits and the risk involved in the drug clinical trials. So, in other words, you cannot force anybody to engage himself in a clinical trial. Whether the patient or the health volunteers willingly must willingly do what? Without any type of intimidation, be able to do what? To sign the healthy, the consent forms. So if you freely and willingly sign the consent forms, that means you are not under intimidation. And before you sign, you have to go through, like I said before, the benefit and the risk. And under the medical ethics, the benefit must outweigh the risk. If you're putting somebody under a clinical trial, your trial must have the benefit that does what? That outweighs the risk. If the risk outweighs the benefit, then the trial cannot be what? Cannot be done. The U.S. Food and Drug Administration or the European Medical Agency or the NAFDAC, these are regulatory bodies. In each country, they have their own regulatory body. So in US, you have the US Food and Drug Administration. In England, you have, in Europe, you have EMA, which is European Medical Agency. And in Nigeria, you have the NAFDAC. These are the regulatory bodies. So if the risk outweighs the benefit, that means the trial cannot be consented. That means the regulatory body will not allow such trial to be performed on human subjects. The benefit must outweigh the risk. So that's one of the things. And the person must freely read the consent form without intimidation, without force, and read it to an understanding where he or she will freely and willingly read it and then understand it and all the benefit and all the risk. And when he understood it, then he will now sign the consent forms for him to be able to participate. And also, where you have a mentally incapacitated patient or volunteer or patient that cannot sign the consent form, because mentally incapacitated patient cannot sign a consent form, then a family member of that mentally incapacitated patient can sign on their behalf. Children can also allow to participate in a clinical trial, but under the guidance of their parents, who will be the one to sign the consent form, because children cannot sign the consent form, especially where the study will benefit the child. But even if the study will benefit the child and the parent signs for the child on their own can decide and say, I don't want to involve in this trial, I am not agreeing to it. You cannot force that child to engage in clinical trials. So that's all about the consent forms. And when they have signed it freely and willingly without any inducement, you're paying them not because you want to bribe them to come in to participate in the clinical trials. You are paying them as you don't have to pay as an inducement. I mean, try to encourage them to keep coming or going to everywhere they're doing clinical trials or to deceive them. You are paying them, giving them money for the inconvenience of having to put them in a place for a period of, let's say, 2 weeks or 1 week and throughout the duration of the study. The inconvenience is what you pay for, and that inconvenience takes about 2 weeks or 3 weeks or whatever, depending on the length of the study. So what am I trying to say here? I'm trying to say that you don't induce, instead you pay for inconvenience. It could be about just £1,000. It could be about $1,000. It could be like about 300,000 naira to engage someone. It could be like $2,000 or £2,000, depending as a payment for inconveniencing the person in the studies. So, and the person will start, and when the studies start properly, we're looking at 10 to about 90 persons in the phase 1 clinical trials who are held. So what are we looking at the clinical trial phase 1? We are looking for safety parameters of the drug. We are now doing what? Dosing. We're doing multi-dosing. and we're dosing. So we want to determine the dose of the drug that will be what, and how the body is going to tolerate this dose. We're looking for the pharmacology. Pharmacology is what the body does to the drug. We're looking at what the body does to the drug that is being taken in. How is your body reacting to the drug? We want to know through pharmacology. Then pharmacodynamics, we want to know what the drug is doing to the body. Remember, pharmacology, what the body is doing to the drug. Pharmacodynamic, what the drug is doing to the body. Now, we look at pharmacokinetics. And in the pharmacokinetics, we're looking at disintegration time of the drug. When you swallow the drug, what happens to the drug? What is the time of the drug dissolving in your stomach? And we're looking at first pass metabolism. And we're looking at whether the liver metabolizes the drug and how the liver metabolizes the drug. We're looking at drug absorption. We're looking at how the drug is absorbed. Were the drug able to pass through the blood-brain barriers? Is it being able to do what? Pass through the blood-brain barriers? How is it going to cross, and how is it going to survive the acidity in the stomach? We're checking all these things because the pH of the stomach is so acidic that some drugs will not be able to be delivered through orally. So that's what we're looking. We're looking at that dissolution. We're looking at drug absorption. We're looking at drug distribution when the blood gets into the drug gets into the blood, and how is it being distributed. We're looking at bioavailability. We're looking at bioavailability. Then we're looking at excretion. How is the drug is being excreted, and so many things involved in pharmacokinetics. So remember, I said that this test is on healthy volunteers who had scaled through inclusive and exclusive criteria. And again, when we have done that and done the dosing, we also said about dosing, then after that, the pharmaceutical company, who is being represented by the clinical research organization, will now, having passed through this, apply to the regulatory body to give a license to do what? To continue on a second phase of the clinical trial. So it is the regulatory body that will license the pharmaceutical company, who is being represented by the clinical research organization or the clinical research associate, to do what? To allow them to go ahead for the participation of the phase 2 trials. And the regulatory bodies have to go through the test result of phase 1. They're going to check the data and check everything, and if it goes well and everything is conducted according to the way it's supposed to be, and it has not gone contrary to the medical ethics, then they will give them a license and permission to proceed to the second phase of the study, which is called phase 2 of the clinical drug development. And in phase 2, we're talking about hundreds, maybe 100, 200, 300, 400, 500 to 900 people were involved in this trial. And in it, we have to use patients. We are not using healthy volunteers. And we look for patients' tolerability to the drug. We're looking at safety parameters as well. Okay? We're looking at how patients react to this, so the safety parameters and also the efficacy as claimed by the pharmaceutical company. Pharmaceutical company claimed that this drug in their protocol will cure a certain disease. So we want to know if the claim of the pharmaceutical company will work in the trial. So efficacy is the maximum response of a drug. Okay? So the maximum response of a drug is the efficacy. So we're looking at the efficacy of a drug. So we're also we'll compare this drug with existing drugs. If it's an antiretroviral drug, if it's an anti-cancer drug, we want to know where are other anti-cancer drugs. How does this one differentiate from the other ones? What is it that our own has that the other one doesn't? We want to look at what our drug is doing that the other ones is like. If you have zidovudine, which is antiretroviral drug, zidovudine, if zidovudine, we are comparing zidovudine with another antiretroviral drugs maybe we have produced, we want to see, and that's where we're going to blind studies. We'll make sure that the investigators or the CRO company does not know the right drug we are using, the new drug. So that's when the blind study where both the investigators and all the people working there does not know which one is what the drug that is on trial. So we'll bring the zidovudine, we'll bring maybe if the name of our drug is called Zelac, we'll bring the Zelac, we'll bring the zidovudine, then we'll bring the placebo. The placebo is the one that is not actually a drug. It does not have a pharmacological effect on you. So, but it will appear in a tablet or capsule depending on how you want it, and the patients will take it. So with that now, because it's a blind study, the investigator does not know what is going on. So it's after that that we'll begin through the data, compare the test result and how effective these drugs were, and we're going to know if the new drug was able to do what? To cure disease more than the other ones, and see how long does it take to do what? To dissolve in the stomach. And look at the plasma membrane bound. How does it bound in the plasma membrane? Is it fast or not? We're looking at the half-life of a drug. Does it have a long half-life? Any drug that has a long half-life is a very good drug. A short half-life drug is what? Is not really good compared to the long half drug. Yeah? So that's what we're looking at. So we're still looking at the existence of the pharmacology, pharmacokinetics, and studies, and is also paramount to our studies. So when this has gone through the stages, the pharmaceutical company, through the clinical research associate or the clinical research organization, will now do what? Because the clinical research associate will pass the message of the data of the results to the pharmaceutical company, who now does what? Apply for a permission, for a license to go for what? Phase 3 studies. So when the regulatory body like US Food and Drug Administration, like NAFDAC, or like European Medical Agency, and so many other ones will do what? When they have gone through it, and the ethics committee goes through, and if they found out, or the local ethics committee, that the data they collected shows that the drug is safe, then they can allow for phase 3 studies. And the phase 3 studies, there are multi-studies. We're looking in thousands. You have 1,000, 2,000, 3,000, up to 10,000, 5,000. It's called a large multi-site studies. So you're doing it in different countries and on different ethnic regions. Why? Because, when you're doing a drug like this, people react to drugs differently. For instance, the way you react to drug now, your body responds to drug is not the same way my body responds to drug. Our metabolism might not be the same thing. That's where polymorphism is involved. And because of this, polymorphism can involve due to ethnic group or genetic inheritance. For instance, 1/3 of Africans also develop what we call a side effect that is called hemolytic anemia when they take aspirin, whereas their white counterpart does not experience it. There are some situations that the white are sensitive to some drugs and develop adverse reactions that black people do not. So ethnicity comes in, and that's ethnic groups, race, maybe white, black, Asians, South Americans, Hispanics react to drugs differently. So that's where polymorphism of drug metabolism is involved. And for instance, in all the studies I told you, I gave you an example of Chinese people, how they react to a very small percentage of alcohol, get them intoxicated because they lack the metabolizing key enzymes that metabolize alcohol. Whereas a Nigerian man or a British man or American man can take 2 pints of beer and will not get intoxicated because they have the alcohol metabolizing enzymes in them, where the Chinese people shows absence of these alcohol metabolizing enzymes. and will do what? Will get intoxicated easily. So when you give a Chinese man a cup of alcohol, a cup of beer, when you finish it, you get intoxicated. Whereas a Nigerian man can take 12 bottles of beer and still be asking for more. This is where polymorphism does. So it's also drugs. So you have to test in different people. The reason why you're going worldwide, you're going to some countries, you're going to test in South America, you're going to test in North America, you're going to go to Pakistan, go to the Asian countries, the Chinese people, you come to Africa, you come to Nigeria, you come to Ghana, or Sierra Leone, or all these places, or South Africa. The reason why you're doing these things is because you want to see what this drug can do to these people of different race. So you cannot conclude because you did it in England, you can't test in England or in America, and you have American population or English population, you think that has represented the whole world. And you will think that that will cure all diseases without side effect. No, because you have to go beyond Europe, you have to go beyond Asia, you have to go beyond America, you have to go to Africa and some other countries to test how these drugs develop. Like I said to you, we are all created the same, and we have the same genome. But about 1% of the entire population of the world do show absence of some key metabolizing enzymes. And they lack these metabolizing enzymes, and they react to drug differently. Some do not get the effect of the drug or the efficacy of the drug. So they don't get relief. For instance, in other studies I told you about codeine. I said to you, codeine is a drug that you use to do what? To take away pains, relieve pain. But some people will do what? Will have relief when they take relief of pain. That means their pain will disappear when they take the codeine. Because of what? They have the key enzyme that metabolizes codeine and convert codeine to its active form, which is morphine. It is the morphine that gives you the relief from pain. So it's not just the codeine you're taking. There's an active thing there that's called morphine that takes away the pain. So the people that lack that the codeine key enzymes does not get relief. They get less relief. So when they take codeine, say people that has the metabolism, they say, oh, when I take codeine, I don't feel no pains anymore. And that's it, but I take the same codeine and it does not do anything for me. It does not do anything for them because of what? They don't have the key metabolism enzyme. So these are how human beings are, and that's why they have to do large trial to find out what this drug does. When they have done it and have collected the data, they will now do what? Will now take it and apply to the regulatory body, the FDA, like I said to you, or the EMA, or the NAFDAC, for a permission to do what? To go into post-marketing surveillance. That's a phase 4. Because by this time, the pharmaceutical company might have spread money through their nose. They spend a whole lot of money. To go to different countries and conduct large studies like this involve a lot of money. Now, the pharmaceutical company wants to do what? To reap from where they've sown. They want to make money now out of it. So they're going to send the data from here to the ethics committee or the ethics regulatory body, who will now grant them the permission to go to this. So they now move from phase 3 to phase 4. You get what I'm saying? So phase 3 to phase 4, now that ends phase 4 of the trial, which is called post-marketing surveillance. Then they start to market the drug. The drug comes in the markets. And now, but they have to contract the physicians, the doctors, the medical practitioners, the GP, all around part of the world to do what? To still monitor these drugs. Sometimes you can see a drug that has been in the market for a period of, let's say, 8 years, 12 years, and all of a sudden, it is discovered that this drug is causing havoc, it's killing people in some parts of the world. When people take it, they die, or they show some serious adverse effects. And once it is reported, the pharmaceutical company will do what? Will take it off from the market. So they now contract these physicians to monitor their patients, the symptoms they have. If they have adverse reaction, if they have side effects, is it a serious side effect? If you're my patient and I'm a doctor, and I'm now doing a surveillance for this particular drug, and I give you the drug, I try to dose you maybe 2 times, 3 times daily, I give it to you. You come back. I tell you, come back next week and tell me how you feel. You come back telling me that since you gave me this job, I have not found myself. It's like I'm not seeing anymore. I'm getting blind, and I can't breathe well. My heart is thumping. It's like I'm having a heavy chest pain and serious side effect that is more than the therapeutic effect of the drug. So the more people are reporting, the physician is reporting back to the pharmaceutical company. And in that way, the FDA, because the report is going to them, will withdraw the drug from the market. This is how we go to do what? To evaluate and also make a market surveillance on the drug, even though they are making money. But if the act of making money will bring death, some people can take the drug and they die. So if they are reported death and people took it and died, it has to be taken away from the shelf. So do you wonder why in Africa here, when I came back from London, I was asking, where are the chloroquine and the nevirapine I used to? They said the chloroquine has been withdrawn from the Nigerian shelf. And the nevirapine was, I said, why was it withdrawn? They say, maybe because it was given some unpleasant, we have scratch and rashes, and people begin to scratch when you take it, some unpleasant side effects and all that. That's why now, or some other things that make them to take it off the shelf of the pharmaceutical market in Nigeria. So that's brings us to the end of this lecture. But before I conclude finally, I just want to summarize clinical drug development. I say clinical drug development is the development of drug. That's clinical trial of the drug. As you do the drug clinically and involve human experimentation. And I said before you go into human experimentation, you must have gone through the toxicological animal dosing. And it is when you get through this that you enter clinical trials. And I also talked about inclusive and exclusive criteria. I also talked about the consent forms. The benefit must outweigh the risk. If the risk outweighs the benefit, the trial will not go ahead. If the benefit outweighs the risk, the trial will go ahead. I talked about phase 1 clinical trials. That is between a small population of people, between 10 to about 90 or 80 or 30 or 50 people, healthy volunteers. And I say a healthy volunteer is somebody who has not got an underlying sickness or medical condition, and somebody who is healthy enough. And I also say that it could be inclusive, that this person is healthy, and the inclusive criteria could be that we want to male of certain age, maybe between the age of 20 to 30 years, or between the age of 20 or 18 to 50 years. Depending, it could be exclusive. So anybody above that age range or below that age range is now under exclusive criteria. That means the person is excluded. Okay? If it's a men that are required, then women are exclusive criteria. If it's women that are required in trial, it is men that are exclusive criteria. I said that. And then that brought us to what are we looking in phase 1? We're looking at pharmacology, pharmacokinetics, thermodynamics. And I said pharmacology is what the body does to the drug. I also say pharmacodynamics is what the drug does to the body. I also told you about what pharmacokinetics is, and I talked about drug distribution, absorption, and all that things in plasma membrane. I also said about the health volunteers, why they are involved and how they are being screened. Now, and I also said to you that phase 2 can only be permitted if all things went well in phase 1. So phase 2, we're looking for patient tolerability, and safety 2, we're looking at the efficacy as claimed by the pharmaceutical industry. We're also comparing with placebo and other drugs, existing drugs, okay? The drugs, and we're looking at pharmacology, pharmacokinetics, and all that problem. We also say that if everything had gone well, that the regulatory body will also grant the license to do the lab studies, phase 3 studies. And I said phase 3 studies between 1,000s. 1,000 could be 2,000, 3,000, 4,000, 5,000. It's called large multi-studies on different ethnic groups. We talk about ethnic groups, it could be Asians, it could be African, it could be American, and North American, South American, and all that. And it's just to do what? To make sure we know what is called polymorphism of drug metabolism. We want to know how the whites react to this drug, how the Asians, how the South Americans, how the Hispanics in other parts of countries respond to this drug. We also thought when they have granted them license, if everything has gone well, then we'll enter into phase 4, which is called the marketing surveillance of the drug. And I said during the marketing surveillance that the doctors also follow this. This comes to the end of the the lectures. And I'll advise you to subscribe to my channel, Timothy Okwebelem, and you can click on the bell, and that will also make you to sure that you see all my videos. Thank you very much for your time and for listening and for watching. Yes, bye.